EurofinsDiscovery provides, within its Translational Biology Center of Excellence, 4 main areas of study. And all of these areas encompass different aspects of translational cell biology. We have a platform called the Biomap platform, and I will talk about each of these platforms in subsequent slides in more detail. But the Biomap platform takes advantage of human primary cell types to accurately and translationally model different aspects of human tissue biology and disease. We also have a biomarker services platform, which can be used to measure different biomarkers in a variety of cellular systems, as well as in a variety of biological samples. Again, I will describe that in a little bit more detail in subsequent slides. We have the OncoPanel service, which is really a cell biology service that is dedicated to looking at anti-cancer agents, and specifically direct anti-cancer agents. And we'll talk a little bit more about the OncoPanel service and how that is set up and used. We have also recently, as of last year, acquired a small company called Discovery Biomed, and that is now set up as our Translational Cell Models department. And this is our newest part of the Translational Biology Center of Excellence. And Translational Cell Models also uses human primary cell and tissue systems that are derived from human samples, both for normal and diseased tissues. So it exploits the power of human tissue biology to provide solutions for our clients. And then for all of these services, to different degrees, we can provide customized services. And we can talk a little bit about that as we go through the different platforms available. So to start with, I will tell you about the Biomap platform. Biomap is a cellular phenotypic platform that takes advantage of the ability to co-culture different human primary cell types together to model in vitro different aspects of human tissue biology and disease. There are a variety of different models available. We call each model a system, and each system models a different aspect of human tissue biology. For example, we have systems that model T-cell activation. We have systems that model fibrosis, and we have systems that model a variety of other different disease and tissue types. With almost all of our models, we use only human primary cell types. I say almost all because we do have some models or some systems that model the tumor microenvironment for those people studying immuno-oncology. In those cases, we use human primary cells with the addition of a standard human tumor cell line. We can provide a brochure that gives more detail on the different systems, how they are set up, what cell types they use, and what biomarkers are measured for each system. We have an extensive database and data analytics tools that can then be used to evaluate the profile for a test agent that is generated in a Biomap study. And we can present the data in a variety of different ways. As a consequence of that, we can use those data to identify a variety of different parameters relating to the test agent. This includes looking at things like mechanism of action. If you have a test agent that is, for example, identified from phenotypic screening, but the mechanism of action is not yet known, we can evaluate that using Biomap by evaluation in a number of different systems, creating what I always call a molecular phenotypic fingerprint based on the profile, and I'll show you that in the next slide. And we can compare that profile with our database to see if it looks like any other agents we've tested, and thus give information on the mechanism of action. We can also evaluate things like selectivity, and we can also guide, in some cases, potential indications. For example, a test agent may have unknown properties that it turns out are actually useful from, say, an anti-inflammatory perspective. So we can really tell a lot of information about a test agent by its profile in a biomap experiment. We can do a number of other things, and I'll just highlight a couple here. For example, we can look at toxicity signatures. There are obviously a lot of in vitro safety pharmacology and toxicity assays, but the complexity of biomap is such that we can identify those molecular phenotypic fingerprints that in certain cases might indicate a potential toxicity alert. And we have a number of toxicity profiles with which we can compare a test agent. We can also do combination studies in biomap. So again, we can evaluate each single agent alone and then in combination to see the benefits, or in some cases, the disadvantages of potentially combining those two agents. What makes the biomap platform tremendously powerful is a huge database that we have, where we have previously evaluated over 4,500 different benchmarks. These might be agents that have been approved as drugs. They may be agents that have been tested in the clinic and for various reasons failed. And even those agents can provide valuable information. For example, knowing how they failed and if it related to a certain toxicity gives us information that we can then use as part of our data analytics. We also have a number of reference agents, and I should mention also that our agents include both small molecules and large molecules. So this large database gives us a tremendous ability to compare a molecular phenotypic fingerprint in biomap with a wide range of agents that have been previously tested. And this is how we can identify many of these parameters in the middle box here. This slide shows a typical trace or phenotypic fingerprint that we generate from what is called our Diversity Plus offering. This is typically an entry-level offering for biomap. And in Diversity Plus, we will evaluate a test agent against 12 different systems, each system here with a name and being represented by the little cartoon. So for example, MyOF here is one of the fibrosis systems that we have. So each system models a different aspect of human tissue biology and disease. And on the bottom here, I don't expect you to read this, but each system has a number of biomarkers that are relevant to that biology. So for Diversity Plus, the agent is tested across 12 systems, and we measure 148 different protein biomarkers, as well as any indications of the test agent's ability to cause proliferation or to result in cytotoxicity in its readouts. So this is what I describe as the molecular phenotypic fingerprint. Now, there are several things I should draw your attention to here. We test agents over 4 concentrations, so we can get an idea of how the agent looks at different concentrations from high to low. We also compare with the baseline or the background readout for each biomarker. So this is shown in the gray signal in the back here. So you can see for the BT system, for example, there is a typical variance in the background signal for some of the biomarkers, whereas in some of the other biomarkers, there's a much tighter window of background signal. So any compound or test agent induced change, as we can see here by these peaks, these are significant deviations from the background that are representative of true biological changes with respect to the biomarkers that are measured. So with this profile, we can see how this profile compares with other profiles in our database, and this is how we can identify things like mechanism of action. We can also compare the profile of a test agent with standard of care agents. If, for example, you have an agent for which you are planning to submit an IND, we can evaluate your test agent here against the same standard of care agents with which your test agent would be compared in a clinical trial. And we can look to see whether there are differentiating factors that as part of the IND submission may be advantageous to indicate where you are showing, at least in this in vitro study, where there may be expected to be benefits or differentiating factors in your clinical agent for which you're applying the IND. Now, as I'd mentioned, we can use those profiles also to identify if there are potential toxicity risks. We have 9 different toxicity signatures, and each signature has standard or hallmark patterns with respect to those different biomarkers that relate to the different toxicities. So each of the toxicities are outlined here in these boxes, and there are examples of reference drugs that are known to cause these toxicities. And then just below that, there are pathway targets that potentially could be resulting in those toxicities. So for example, here for skin rash, typically something like a MEK-mediated toxicity. This relates to MAP kinase pathway biology, and so some of the targets are shown here, and drugs relevant to that are shown here. So again, by comparing the test agent profile with known toxicity signatures, we can evaluate if a test agent may be expected to result in a toxicity. Obviously, if this is done earlier in drug discovery, then through, in the case of small molecules, chemical modification, that toxicity can be dialed out of the profile at an earlier stage. This is an example here for telmisartan. So these are the 9 toxicity signatures, and this is the result we see with telmisartan when tested over a range of concentrations. And you can see here, in some cases, there is no detectable toxicity, but in other cases, there are toxicities either seen at very high concentrations or somewhat lower concentrations. For example, skin sensitization here at concentrations greater than or equal to 3.3 micromolar. So telmisartan is an approved drug, and just because a toxicity is seen doesn't mean to say that that compound will not move forward, but it allows you to see whether that toxicity might be manageable. So this is a good example of where we can use biomap in this particular case. I apologize for that. There was a slight delay in the slides advancing there. So this slide shows the range of different services we can offer within the biomap platform. I had mentioned the Diversity Plus panel, and this is typically an entry-level panel to provide that initial profile for a test agent. But then there are sub-panels. For example, if you see interesting biology that relates to T-cell biology and autoimmune biology in an inflammation setting, we can evaluate the test agent further in a panel of models or systems that relate to that biology. Similarly, we have things like the fibrosis panel, and then I'd mentioned the immuno-oncology tumor microenvironment panels here. So these can give more detailed evaluation of a test agent's profile with respect to biologies of interest. We can also conduct what is called early screening, or as we call ES. So we can do this with diversity early screening. And in this case, you can evaluate many more compounds in a more cost-effective manner. We provide a similar report to Diversity Plus. And in that case, it is a slightly simpler report, but still allows actionable decisions to be made from the profiles that are generated. We can also use the platform to triage compounds or test agents in an early screening format, and also in a systems-based format. There are a number of additional services that can be elected as add-ons. We can use System Elect. For example, if you just want to look at one system, we can do that. If you indicate the system of interest, we can use that in the study. We can then do combination studies. For example, an elected combination in a particular system. And we can, in certain cases, look at the effect of the test agent on RNA levels. As I'd mentioned, we have toxicity signature analysis, which is an add-on to typically Biomap Diversity Plus, and a variety of other data analysis packages as well as overlaying a profile with a known agent, again, to see where there are similarities and differences between the two. So that is the Biomap platform. Switching now to OncoPanel, as I mentioned, OncoPanel is an in vitro cellular phenotypic platform that is designed to look at the effect of agents from a direct anti-cancer perspective. This platform takes use of a large number of human tumor cell lines. Now, I say that this is useful for looking at direct anti-cancer agents because all of these cell lines are cultured in homogeneous culture. They are not cultured with other cell types. As I've mentioned earlier for Biomap, if one is looking at immuno-oncology agents where an immune cell component is needed, the Biomap platform is better for looking at that. But this is looking at the direct effect of agents on the viability and proliferation of human tumor cell types. We have a wide number of different tumor and tissue lineages represented. And what makes our platform very powerful is we have a genetic database where we have over 270,000 different genetic aberrations and features represented. So in certain cases for larger studies, we can correlate drug response with genetics of the tumor cell lines to identify predictive genomic biomarkers for both sensitivity and resistance to that test agent. We have used this platform for a large number of clients with a long period of experience. We have a lot of people on the team, myself included, that have backgrounds in big pharma with oncology drug discovery. In addition to 2D models, we also have a number of 3D spheroid models. So these are grown in 3D culture, again in homogeneous cell culture, but these can give an additional indication as to whether a test agent will work in the context of a 3D structure rather than cells grown in 2D. We do have a growing database of reference agents, and many of these are standard of care agents, but also several reference compounds. And as part of a genomic analysis for a larger study, we can also compare a test agent with this database to see whether it looks in its profile similar to anything we have studied before. And we do have flexibility to be able to customize many of these assays, and we're very willing to talk with you about ways in which you might want to customize your study. We would typically do that on a case-by-case basis. So what is the OncoPanel platform then? As I mentioned, it is a flexible platform for evaluating the effect of direct anti-cancer agents. We have more than 300 different human tumor cell lines that are typically screened in a fixed order every 3 months. However, we do have the flexibility to use studies that have smaller numbers of cell lines, anything from 1 cell line to the maximum number available for a given assay format. So we can accommodate lots of different scopes and sizes of study. And for those smaller studies, we are able to start them on an ad hoc basis. So you don't have to wait, for example, for 3 months for a quarterly screening start for a smaller study. We do also have the flexibility to start even large studies in between those quarterly start dates. In that case, there is no change in price, but a study has a very slightly longer turnaround time because it by definition would span 2 screening panels. As I'd mentioned, we do have broad cancer type and subtype representation, and we do have the ability to do, as I'd mentioned, full panel or custom designed sub-panels of cell lines. For example, you might only want to look at breast cancer cell lines, or you might want to look at cell lines that have a particular genomic feature, for example, BRAF mutation. And we can work with you to help design panels of cell lines that best meet your needs in that respect. As I mentioned, we have extensive genomic characterization for the vast majority of the cell lines in our collection. And the data used for this are actually obtained from 2 public access sources. One is the Cosmic Sanger database in London. The other is the Broad CCLE database in Boston. And we have, with an external company that specializes in database integration, normalized and integrated those 2 data sets. So what we have effectively is a proprietary database that represents a fusion of those 2 public access databases. And as I'd mentioned, we can use this to correlate with drug response to identify predictive genomic biomarkers for both sensitivity and resistance. So this can potentially allow you to stratify patient populations for a clinical trial, and it can also identify potential intrinsic resistance mechanisms that can then rationally inform on combinations that might be worth exploring to enhance the utility of your asset. The data that we generate are robust and high quality and are very reliable in terms of providing actionable data. The platform itself is fully automated and uses 384-well format, and we have, as I say, been running this for many years. We have a multi-layered QC process. We have a lot of historical data for the use of all of our cell lines in all of our standard assays. And for every study, we correlate the performance of each cell line in each study with historical data, both at the level of the growth rate of the cells, as well as their response to a known reference agent. In that case, we use staurosporin. And so for a cell line to pass QC, it must be growing at a consistent rate with historical performance and responding to staurosporin in the same manner that we have seen historically. So that means that, for example, if you did a study with us maybe 2 years ago, and then you do a study with us in the coming months, that you have a lot more confidence in comparing particular cell lines between studies for the very reason that we have this multi-layered QC process. We also culture cells at low passage number to prevent genetic drift. So we do not keep cells in culture for extended periods of time. If a follow-up study is needed, we will pull new vials of cells from our extensive banks. And again, all studies use this extensive QC process. So in terms of the assay types that Oncobanol provides, in 2D culture, we have standard assays validated for exposure times of 3, 5, and 10 days, with up to 301, actually now 302 cell lines for 2D studies with 3 or 5 day exposures, and 269 cell lines for 10-day studies. We have several different assay formats. We have a single-plex format that uses DAPI nucleus staining with high content analysis, and this provides a straight proliferation readout. We typically test over 10 concentrations, so this allows us to provide EC50, GI50, IC50, and activity area readouts. And this will give an indication as to whether the test agent has the ability to inhibit the proliferation of the tumor cell lines being studied. We use high content analysis because in our standard multiplexed assay, not only do we evaluate the effect on proliferation, we also evaluate the effect of the test agent on apoptosis by looking at levels of cleaved caspase 3. And for that, we report the test agent concentration required to give a 5-fold induction over the vehicle control. And then we also measure the effect of the test agent on the cell cycle by measuring levels of phosphohistone H3, and again, reporting relative to the vehicle control, whether the agent, the concentration at which it gives a 2-fold increase for G2M arrest, or a 2-fold decrease or 50% reduction in signal for G1S arrest. So the multiplexed assay gives more information on how a test agent performs. I always say the single-plex proliferation assay asks the basic question, do my test agents inhibit the proliferation of human tumor cells? The multiplexed assay goes a little further and asks the question, how do my test agents inhibit the growth of those tumor cells? Now, we do also have a viability assay. This is a standard cell titer glow assay, and this is a straight bioluminescence assay. So it gives a true viability readout. I had mentioned we can perform genomic analyses, and we typically do this on any study with 200 cell lines or more, up to and including the full panels available for use with these assays. And as I'd mentioned, we can conduct customizations of assays, which we would do on a case-by-case basis. I'd mentioned that we do have 3D spheroid assays available. We currently have 117 different models, and these can be customized upon request as well. These are also viability assays, so can be used as effectively a comparison with the 2D viability study. In this particular case, we also report EC50, GI50, IC50, and activity area values. We can do drug-drug combination studies with all of the formats above, and we do these typically with a 9x9 concentration combination matrix. And for that, we generate Bliss scores and a heat map. So you can see where combinations give areas of synergy, additivity, and antagonism. We do also have a panel of normal cell types. Currently, we have 8 different normal cell types from a range of different tissues. Most of these are normal primary cells, and we would typically use these, or a client may typically use these, if in, for example, the 2D screening, one sees very broad, apparently broad spectrum cytotoxic activity. And these can be used to check that the agent is not a general cytotoxic agent. And we can, as I'd mentioned, perform either cell banking or custom studies on a case-by-case basis. I'd mentioned about the genomic analyses, and just very briefly, I can say here that we look at both mutational features as well as differential gene expression. So we can see if certain mutational features correlate with drug response, as well as whether overexpression of certain genes correlates with drug response, both at the level of resistance as well as sensitivity. So this analysis can be very powerful in informing the tumor types in which an agent would be expected to work. So now I'll turn briefly to biomarker services. Biomarker services is a very broad and diverse platform. In a very simple sense, we can take a test agent and we can treat a cell platform. And this can be very wide and varied in terms of the kinds of cell platforms we use. This platform is very highly customizable. So we do have a number of standard catalog assays, many of which look at immunomodulatory activity, but we can conduct assays using pretty much any cell type, as long as either we can procure it or you can provide it. And if we can set that cell type up in vitro to model the desired biology for a particular study with appropriate stimuli if needed, treat with a test agent, and then measure various biomarkers. This may be soluble protein biomarkers like cytokines. It may be phosphorylated biomarkers in an intracellular context. Really, as long as there is a reagent available to detect that biomarker, we should be able to set up a study for you. Now, we can also conduct studies where, for example, the cell biology part has already been done. These may be ex vivo samples. For example, if you have done an in vivo study and you have samples of plasma, and you say, can we measure certain biomarkers in those plasma samples? Again, if there are suitable detection reagents, we don't need to do the cell treatment part because that's already effectively been done in vivo. But we can then look at those samples in a non-GLP setting to measure the biomarkers of interest. Now, as a result, because what we can do here is so wide and diverse and so very highly customizable, we actually base our capabilities around a variety of different biologies and in particular technologies. And I'm not going to go through each of these in great detail, but we have a wide range of different technologies that we can use to measure the particular biomarkers of interest. And this can include platforms like the Luminex platform and the Mesoscale Discovery platform in cases where one might want to measure multiple biomarkers from the same set of treated samples. Now, I'd mentioned that the measurement of biomarkers can be done as long as there are detection reagents available. Typically, most technologies use an immunologic detection mechanism, and that would be true of Luminex, of ELISAs, of HTRF assays, of MSD assays, excuse me. But there are certain cases where there may not be a suitable detection reagent. Now, in some cases, we may also be able to employ mass spectrometry. We have world-leading mass spectrometry capabilities. And so there are sometimes opportunities to measure various biomarkers using mass spectrometry of either the treated cell samples or the biological samples that you may be sending us. So this platform has tremendous breadth and scope in terms of what we can do. Simply put, if you want to measure a biomarker in a certain cell system or in a biological sample from a certain cell system, there is a very good chance we could do such a study. We do have some standard assays, and some of these assays look at things like immunophenotyping. We have a range of assays that I'll talk about briefly on the next slide. They can look at how biomarkers change in a variety of different immune cell types. We can look at aspects of immune safety, for example, using what's called a cytokine storm study. And we can also look at immune responses in designated target-based assays, for example, looking at things like toll-like receptors or particular immune cell types. So with respect to working with biomarker services, there are a wide range of different things that we can do to support drug discovery programs at multiple stages, from target validation right the way through to IND. This is just a brief overview of some of the immune modulation assays that we have, and these are typically our catalog assays within biomarker services. So we have a variety of assays to look at the effect of test agents on T cell function, and this is typically using PBMC, human peripheral blood mononuclear cell samples, from a variety of normal healthy human donors. And we can then look at the effect on T cell function by measuring their ability to secrete different cytokines. Each different aspect of human T cell biology, whether it is through activation of the T cell receptor or through the effect of T cell mitogens, such as PHA or CON-A, we can then measure distinct cytokine profiles. And we can then look to see either if your agent stimulates T cells or if your agent inhibits the activation of T cells. We can also look at similar activities against macrophages and monocytes. So in this case, we also use PBMCs, but we look at a different panel of cytokines. And we have, in this case, either a different positive control for the assay or a different stimulation. Typically for the macrophage monocyte assays, we're using LPS, whereas for the T cell assays, we're using a range of other stimuli. As I'd mentioned, we have a number of toll-like receptor assays available. And just like the immune cell type assays, we can set these up either in activation mode or in inhibition mode. And for each of these TLRs, we have different ligands that are specific to each type. And those ligands can either be used as the positive control in an activation assay, or they can be used as the stimulus in an inhibition assay. And again, just like the assays above, each TLR assay will have its own panel of cytokines, which is indicative of activity through that TLR. I had mentioned we have a cytokine storm study assay, and this is particularly important for anybody evaluating or developing biologic agents. I won't go into the history here, but it has been observed previously that biologic agents can potentially elicit a cytokine storm. Unfortunately, over the last 2 or 3 years, I think we've all become very familiar with cytokine storms through the action of COVID-19. And this can happen when any agent that is foreign to the body is introduced, especially biologics, that can elicit an unwanted and uncontrolled pro-inflammatory reaction. So there are a number of cytokines that have been identified by the US FDA that are indicative of a cytokine storm and that are strongly advised to be evaluated with any biologic agent to make sure that that biologic agent does not cause a cytokine storm, or at least a major cytokine storm. So we can set up studies here either to look at very simple evaluations of perhaps multiple test agents with maybe 1 or 2 PBMC donor samples, right the way through to IND enabling studies where we can look at typically upwards of 9 different normal healthy human PBMC donor samples, and we can measure the panel of 9 different cytokines that are indicative of the cytokine storm. Now, in addition to measuring the ability, or hopefully not, of a biologic test agent to elicit a cytokine storm, we can also look at pro-inflammatory drugs. If there are drugs that are designed to boost the immune system, you obviously don't want them to boost the immune system too high. And so in certain cases there, if an agent is being developed, for example, to provide a better immune response as in the case of infection or sepsis, it might be also worth testing that, even if it's a small molecule, in the cytokine storm study. Obviously, cytokine storm can be modulated not only at the mechanistic level by a test agent through activation of T cells and other immune cell types, but also through potentially cytotoxic or pro-proliferative effects. And we can often run a parallel proliferation or viability assay alongside the test study in cytokine storm, so that if you see no response or a minimal response, you can make sure that that's not because your agent has killed all the T cells. So a very useful assay there for looking at immune safety. So now I'll turn to the final of the 4 different parts of translational biology. And this is our newest department. This is our Department of Translational Cell Models. And as I'd mentioned earlier, this takes advantage of the ability to procure normal and diseased human tissue and cell types, and to use that to evaluate the effect of test agents in a variety of different biological contexts. This is just a quick summary of some of the scope of things that can be studied. And like biomarker services, a lot of the studies in translational cell models take the form of customized assays. So, like I'd mentioned, we take the advantage of looking at primary human cells derived from tissues, both diseased and normal. We can conduct 3D and 2D bioassays, and I'll give an example of one of those that we have in a couple of slides' time. The ability to look at normal versus diseased cell types can give a direct comparison of a test agent as it relates to the disease of interest. So this is a very powerful aspect of the translational cell models platform. And as I'd mentioned, many of these studies by definition are customized formats. So again, we would invite you to talk with us about the kind of study you would like to set up, and then we will evaluate the feasibility of that and put forward a proposal for the study that we can do to evaluate and to set up a study that meets your needs. There is also potential synergy of translational cell models with a variety of other different services, including the Biomap, biomarker services, and OncoPanel translational biology services I've mentioned in this particular webinar, but also potentially with our ADME-Tox, ion channels, and biotherapeutics divisions. For example, you might want to see whether a particular test agent permeates into a tissue, and when it permeates into that tissue, if it has the desired effect on the biology in that tissue. Obviously, we can measure the effect here through a biomarker type approach, but we can also measure how much of that test agent gets into the tissue using a typical ADME type study. That's just a simple example of the kind of synergy we can use. We have a variety of different tissue types that can be studied. So some of these are in established models, others are more exploratory in nature. And again, we would invite you to speak with experts that can set up these models. And all of these customized models, just like the customized biomarker services models, typically the way we would run these is that we would set up a feasibility study to show that we can model the biology of interest, we can get the appropriate signals at the level of particular biomarkers of interest, and that we can measure those biomarkers. And if necessary, in the presence or absence of a tool or a reference inhibitor or a stimulant. If everything looks good from that perspective, we would then go to the main phase of the study in which we would test your test agents. This is just some more examples of different customized opportunities that we have. And I think really what you can see from this is that we can look at a wide variety of tissue types from the human body. And our experts in translational cell models have access to a wide variety of different tissue banks from which we can get those primary tissue samples.